Welcome. In this session, we are going to look at the catabolism of branched chain amino acids. So there are 3 branched chain amino acids as shown here. Yes, as you can see here, there are 3, only 3: valine, isoleucine, and leucine. So those 3 are your, so this valine, there, this leucine, and isoleucine. Those 3 are your branched chain amino acids. As you can see, the R group is branched. So you, like in isoleucine, you can see this is the branch. So these are the branches in leucine, and in valine, these are the branches. I want you to see the difference between valine and leucine. So in valine, you can see here it is only the one CH. Here there is an additional CH2 and methylene group. So that is the only difference between valine and leucine. So you should be able to differentiate those two in case you are asked to draw them or something like that. So today we'll be looking at the catabolism of those 3. So let's get to that immediately. So the catabolism of these 3, they follow a similar pathway. Okay, so the 3 of them. So you can see the 3: leucine, valine, isoleucine. We have some color codes here. Leucine in green, it is strictly a ketogenic amino acid. Valine in red is a glucogenic amino acid. Isoleucine in that color, which I might say is brown. Sorry for that. I don't know which color is that, not for sure, maybe brown. It is both ketogenic and glucogenic. So leucine is strictly ketogenic. Valine is glucogenic. Isoleucine is both. So they will undergo some reaction. So they will be transaminated. The 3 of them are transaminated by an enzyme, which is known as this enzyme, the branched chain amino acid aminotransferase. We will transfer their amino group to alpha ketoglutarate, and after transamination, they will form their respective alpha keto acids. So the alpha keto acid of leucine is this alpha keto isocaproic acid. The alpha keto acid of valine is alpha keto isovaleric acid, and the alpha keto acid of isoleucine is alpha keto beta methyl valeric acid. So all these 3 are the respective alpha keto acids of the 3 amino acids. So what next? So after formation of the alpha keto acid, I've said the reaction is catalyzed by the branched chain amino acid aminotransferase. We will form their respective alpha keto acids. These alpha keto acids undergo oxidative decarboxylation. Oxidative decarboxylation, this reaction is catalyzed by this enzyme, the branched chain alpha keto acid dehydrogenase. This branched chain alpha keto acid dehydrogenase is synonymous with the pyruvate dehydrogenase complex enzyme. Also, remember we said I've made a video on the pyruvate dehydrogenase complex. I will attach the link in the description if you want to see it. Remember that enzyme is also synonymous with alpha ketoglutarate dehydrogenase. In what ways? It is a complex of 3 enzymes. And those 3 enzymes, they require specific coenzymes. Yes, one coenzyme which is required by the first enzyme of that complex is thiamine pyrophosphate, a derivative of thiamine, vitamin A. We have CoA. Lipoic acid is required by the second enzyme in that complex. And we have flavin adenine dinucleotide, which is required by the third enzyme of that complex. NAD comes in to oxidize, to reduce the oxidized FAD. You should look at that in the pyruvate dehydrogenase complex. So whatever the branch chain alpha keto acid dehydrogenase enzyme will do, its reaction mechanism is similar to that of pyruvate dehydrogenase and also alpha ketoglutarate dehydrogenase. So if you have not seen that video, please make sure you look at the description to see it. So after decarboxylating, after carrying out the oxidative decarboxylation, we will form their respective acyl CoAs. So from after carrying out oxidative decarboxylation, say of alpha ketoisocaproic acid, the alpha keto acid of leucine, we will form its acyl CoA, which is isovarenyl CoA. So all these are just acyl CoAs, such that they are, even you can see it is the same as acetyl CoA. The same way we were carrying out oxidative decarboxylation of pyruvate, we form acetyl CoA. The same applies here. We will form the respective acyl CoAs. Now, these acyl CoAs will undergo further dehydrogenation. Now, after forming these acyl CoAs, we will follow those steps we were following when carrying out the beta oxidation of fatty acids. We have already formed the acyl CoAs. So remember the first reaction we were carrying out. So this, after forming the acyl CoAs, we will carry out dehydrogenation catalyzed by acyl CoA dehydrogenase enzyme. Acyl CoA dehydrogenase enzyme is FAD linked. Always remember that it is FAD linked. And remember, when FAD goes to the electron transport chain, it will yield two ATPs. So after carrying out their dehydrogenation, you can see here. Valine is this pathway, the one to your left. The left is the metabolism of valine. Valine will form beta methyl propionyl CoA. Those are just minor details. Then we carry out the carboxylation. But I want you to note that at the end of the day, sorry, what did I say? The one on your left, let's go back, is leucine. Leucine, as I said, is strictly ketogenic. So at the end of the day, it will give you these two, acetoacetate and acetyl CoA. Remember, those two will go to form your ketone bodies. That is why it is ketogenic strictly. Okay, that is leucine. But I want you to look at the other ones, the valine and isoleucine. Now, can you look at the valine? Valine here. Valine, after forming its acyl CoA, which is known as isobutyryl CoA, and undergoing dehydrogenation, it will form propionyl CoA. It will eventually form propionyl CoA. Propionyl CoA can be converted to succinyl CoA. I will be showing you that later. So that is why I've said valine is strictly glucogenic because it will only form propionyl CoA. Okay, then let's look at isoleucine. Isoleucine, on the other hand, I've said is both, and I will show you why I've said both. Isoleucine, after forming its acyl CoA, which is the alpha methyl butyryl CoA, and undergoing dehydrogenations, it will give rise to two products, as you can see here. Two, acetyl CoA, which will go to ketone body synthesis, so it is ketogenic, and at the same time, it will also form propionyl CoA, which is glucogenic. That is why I've said isoleucine is both ketogenic and glucogenic. Now, after forming propionyl CoA, how will we obtain succinyl CoA, which will enter the gluconeogenetic pathway? Gluconeogenesis is the synthesis of glucose from non-glucose precursors, if I can say so. So how will this propionyl CoA form succinyl CoA? Let me take you back to some trip. Here we are. Now, this here is your propionyl CoA. Okay? Propionyl CoA is carboxylated by propionyl CoA carboxylase here. A biotin requiring reaction. Remember the reaction which require biotin? Pyruvate carboxylase, and I think also there is acetyl CoA carboxylase. I think those three require biotin. So, propionyl CoA, after being carboxylated, so we are adding a carbon group. So after being carboxylated, what will we get? We will get D-methyl malonyl. D-methyl malonyl. Why D? Can you look at this methyl group at carbon number 2? Here. It is on your right, dexter position, D. D-methyl malonyl. Then we have to move that. Then the other enzyme cannot act on the D form. It has to act on the L form. So we have to carry out isomerization. So you can see this. Maybe I should reduce this to there. Yeah. You can see the D from this position, D position, to the R. Okay. So from D-methyl malonyl to R-methyl malonyl, reaction catalyzed by methyl malonyl, CoA restless. A restless. It is like an isomerase, but call it a restless. So it will transfer that methyl group from the D position to R position. So after doing that, after forming now the R-methyl malonyl, what next? The last reaction. The R-methyl malonyl, I want you to see this structure very carefully, and I show you what goes on. Now this is your R-methyl malonyl. A reaction catalyzed by the methyl malonyl CoA mutase, we will form succinyl CoA. Now that reaction requires vitamin B12, your cobalamin, deoxyadenosyl cobalamin. Now, people with deficiency of this enzyme coenzyme, the vitamin, not necessarily deficiency. Let's say they are peptic cells in the gastric mucosa are not producing the intrinsic factor, which is required for absorption of vitamin B12 in the small intestines. Now, if you have deficiency of B12 in your body, you see methylmalonyl CoA mutase will not be effective. So you will have accumulation of methylmalonyl. So that will be known as methylmalonyl acidemia. Methylmalonic acidemia. So because methylmalonyl is an acid as per se. So, but we look at that in a few. So I hope you have understood how we convert propionyl CoA to succinyl CoA. Those 4 reactions, you must understand them. Let's repeat them. Propionyl CoA is converted to D-methylmalonyl CoA by propionyl CoA carboxylase, a biotin requiring reaction. Then D-methylmalonyl is converted to L-methylmalonyl by the racemase. Then L-methylmalonyl is converted to succinyl CoA by the mutase, which requires cobalamin, vitamin B12. Now, after that, we have formed remember propionyl CoA was generated from isoleucine and valine. So we have formed your succinyl CoA. After forming your succinyl CoA, now it is one which will enter the gluconeogenetic pathway to give you glucose at the end. Now, I want us to look at something short, the sum of the, what if these enzymes are defective? What if they are? Now, let's look at this as the final thing. Remember this image. If you have been watching my videos, you must know this image. Now, we are here. We are here, branched chain. You can see isoleucine and valine. Also, leucine should be here, but this, I will show you where it is. First, we said we carry out transamination to form their respective alpha keto acids, which will undergo oxidative decarboxylation reaction catalyzed by the dehydrogenase, the branched chain alpha keto acid dehydrogenase to form their respective acyl CoAs. Now, what if you have deficiency of that enzyme? You will have this disease, the maple syrup urine disease, due to deficiency of the branched chain alpha keto acid dehydrogenase. So there will be accumulation of this, the alpha keto acids and also the respective amino acids. So in the plasma, in the urine, so you have neurologic problems, high mortality rate. So treatment, to treat this, you can dietary restriction of foods that have the branched chain amino acids. So don't take them. Why take something that will kill you? Don't. So then we said they will undergo, this acyl CoA will undergo some dehydrogenations and they will give you propionyl CoA. Then propionyl CoA is converted to succinyl CoA as I have shown you. Then I said here, from this should be L-methylmalonyl CoA. If you have deficiency of the enzyme methylmalonyl CoA mutase or the adenosyl cobalamin B12, you have accumulation of methylmalonyl, which will lead to methylmalonic acidemia. Okay. So, and it is accompanied by metabolic acidosis and developmental problems. Okay. So those are some of the inborn errors of metabolism that you can have. So let me show you where you see this is where leucine is. So you see, if you have also the deficiency of the branch chain alpha keto acid dehydrogenase, which will convert, for in our case, the alpha keto isocaploid to isovalyl CoA. If you have deficiency of that enzyme, you will have maple syrup during disease. So, but I want to see that leucine will give you acetoacetyl CoA, which will give you acetyl CoA. So leucine is strictly ketogenic together with the lysine. So leucine, lysine, strictly ketogenic. So I think we have come to the end, and thank you guys for your participation and support. Subscribe for more med content. Thank you.